So I am trying to, tell a little story how we moved from cardioprotection to cardiotoxicity recently, and learn together with you from your questions as well, because the cardiotoxicity field is also new for me as well. And I will give you quite a simple presentation for about 30 minutes, and I'm happy to discuss with you afterwards. So here, oops, I try to change. Yeah, okay, this is great. So as the ISHR community knows it well, ISHR originally was the so-called Ischemia Reperfusion Society. So most of our colleagues are aware of this story of ischemia reperfusion injury causing myocardial infarction, dysfunction, arrhythmias, and then heart failure. And after the discovery of preconditioning, which is brief periods of ischemia before the lethal ischemia was shown to exert a very robust cardioprotective effect, which manifests as reduction of infarct size, improvement of cardiac function, and reduction of arrhythmias. And afterwards, the discovery of postconditioning, when a staged brief reperfusion after ischemia may cause also cardioprotection, gave a real birth to this field, because this can be used clinically as well to do a staged reperfusion at the catheter lab in the heart unit. And of course, remote conditioning strategies, also early 90s, where they showed that these brief periods of ischemia can be exert any time in any other tissues around the lethal ischemia reperfusion injury. For example, by making ischemic any extremities like the arm, and that can cause cardioprotection. This was really, very encouraging and motivating times when everybody thought that this can be translated into the clinic very soon, and then we can forget about myocardial infarction, because especially preconditioning causes a robust cardioprotection. However, then the problems came, and the major problem was that especially comorbidities disrupt this very, the nature's beauty, endogenous cardioprotection. Most of the comorbidities inhibit the effect of this cardioprotection. And we were the lucky ones who showed it first in hyperlipidemic animals, hyperlipidemic rats in the early 19s, that this hyperlipidemia disrupts preconditioning. So that was the major funding of our field, that we were experimenting and doing several projects, especially focusing on hyperlipidemia and diabetes and kidney failure, on how this comorbidity might affect both ischemia reperfusion injury and types of cardioprotection. Since then, it has been shown also in small clinical trials and in many preclinical research studies that aging, for example, aggravates ischemia reperfusion injury and decreases, attenuates cardioprotection afforded by either preconditioning, postconditioning, or remote conditioning. Also, hyperlipidemia does the same, so it inhibits pre, post, and remote conditioning in many of the animal models, and it has been proven by many people, many groups all over the world. The same happens with diabetes and interestingly kidney failure, although it increases ischemia reperfusion injury. In some of the studies it's mostly neutral, but still there's very little is known about the effect of comorbidities on the effect of cardioprotection. Of course, in the clinical studies, in most of the clinical studies, post-conditioning and also remote conditioning fails to improve the outcome of myocardial infarction or patients with cabbage. and therefore somehow it's lost in translation, and this is now also a hot topic of cardioprotection. The other problem is that not only comorbidities interfere with cardioprotection, but also the medications used to treat comorbidities. This table was first reviewed in 2014. There might be some newer data, but today I will focus on cardiotoxicity, how we come to this field. But this shows this summary slide that, for example, although nitrates, which is, a drug used more than 100 years for now to treat myocardial infarction patient and angina patients, the development of nitrate tolerance, which is the most common limiting factor of the clinical use of nitrates, nitrate tolerance not only worsen ischemic reperfusion injury, but it also has been proven in some clinical and non-clinical studies to decrease the protection by pre- and post-conditioning. And also, some studies also showed that statins, which are the widely used anti-hyperlipidemic agents, which has also some vascular protective effect, although it decreases ischemic reperfusion injury, but it also might interfere with both pre- and post-conditioning, depends on acute or chronic treatment. So I don't want to go into details to discuss these tables, but there's also some other drugs which would be very familiar with you, that for example, the ATP-sensitive potassium channel blockers, which has been used as anti-diabetic agents, were shown to decrease both pre-post conditioning and also the remote conditioning effect. And also, I will later discuss some of our very recent results on COX inhibitors, the cyclooxygenase inhibitors, which has been shown to decrease myocardial ischemic reperfusion injury, but it has been shown also that it might interfere with pre- and post-conditioning as well. So I will show only two very old data from our lab. So after looking at hyperlipidemia, we showed that hyperlipidemia, blocks pre- and post-conditioning in rat models and also in small clinical trials. We thought that it might be, if we use healthy rats with pre-conditioning and post-conditioning and measure myocardial infarct size, you see the robust effect of pre- and post-conditioning here. It's in isolated rat hearts. Afterwards, with the pre-treatment of chronic statins, we might be able to further reduce infarct size. However, what we received, the results was very unexpected. Namely, that we saw that chronic statin pretreatment decreased infarct size, but it had an antagonistic effect of the post-conditioning effect. So in fact, this chronic statin treatment in this rat model blocked post-conditioning. So it was a surprising finding. And also, we used a post-conditioning model in a normal and nitrate-tolerant rat. And nitrate tolerance was induced by 3 days of treatment with high dose of nitroglycerin. And what we found is that in nitrate-tolerant animals, also nitrate tolerance especially blocks the cardioprotective effect of post-conditioning. So if we summarize these findings, so what we saw is that an ischemic reperfusion injury can be either, decreased by cardioprotective methods like ischemic conditioning or some cardioprotective drugs only in preclinical settings, because unfortunately we still do not have any cardioprotective drug in the market which might reduce myocardial infarct size. And at least we think that this is partly due to the comorbidities in patients and the co-medications of patients. And since the preclinical testing of drugs are not done in these settings, the other thing is that some drugs may aggravate ischemia reperfusion injury, and also some comorbidities can aggravate ischemia reperfusion injury, like diabetes or dyslipidemia, and they can also inhibit the endogenous cardioprotection by ischemic conditioning. So, and these drugs, we can say that these unexpected effect of drugs, that they aggravate ischemic reperfusion injury or they block the endogenous cardioprotective mechanisms, this is kind of a cardiotoxicity, which we can say that this is a type of adverse cardiac event, which haven't been checked in any of the preclinical cardiotoxicity screening so far, because in all drugs are checked in healthy animal tissues or healthy animals. So we think that not only cardioprotective drugs should be checked if they work still in the presence of comorbidities, but also every drug might have this so-called, what we call this hidden, we termed it hidden cardiotoxicity, that these drugs might show some cardiotoxicity, but only in a diseased heart. And that's what we called termed hidden cardiotoxicity. We recently published this paper in the European Heart Journal. So since we were totally new in this cardiotoxicity field, we went on to check how this cardiotoxicity is done in the drug development. And we realized that lots of drugs where cardiotoxicity is revealed only in the very late stage of the drug development. Here you see the drug development route. Only at the very late stage in the clinical trials, or even when the drugs are already on the market. And the current preclinical testing, they don't forecast this cardiotoxicity. And the problem is that this unexpected cardiotoxicity accounts for 60% of drug withdrawal from the market and the termination of clinical trials. And of course, this is not very safe for the patients. So we were thinking that how to predict this cardiotoxicity in the preclinical setting. So this cardiotoxicity, what we read about, we think it's a double problem. First, it's a clinical problem because there are still drugs on the market which shows cardiotoxicity as a side effect. Especially oncologists, doxorubicin, for example, or sunitinib, you see that the ratio of unexpected or the existing and known cardiotoxicity can be up to 20 or 50%. And also, for example, the HER2 inhibitor trastuzumab can have cardiotoxicity up to 20%. The other problem is an economical problem because the drugs are withdrawn from the market after investing a lot of money in the development of the drugs, and because it turns out that the drug on the market already is cardiotoxic. And this is, for example, rofecoxib here. I am mentioning rofecoxib as a COX-2 inhibitor because we will go on that thing later. So we further kept reading about the mechanism of cardiotoxicity of the drugs. And it was also another surprise for us that the mechanisms of cardiotoxicity, even known drugs like doxorubicin or trastuzumab, very little is known about it, or it seems that it does everything. So, for example, in this review of my colleague and friend is showing that the doxorubicin drug-induced cardiotoxicity might have a lot of targets, so it often involves oxidative nitrosative modification that leads to mitochondrial dysfunction. So it's a very general issue. And then we realized that there is an old classification of cardiotoxicity. There's a type 1 and type 2 cardiotoxicity. And the type 1 is called the direct cardiotoxicity, where the drug shows a direct cardiotoxic damage. A typical drug is anthracycline, like doxorubicin. And here you see that there's a lot of targets, for example, lipid peroxidation, apoptosis, myocardial damage, and energy depletion, adrenergic dysfunction, and so on. So a lot of mechanistic effect. So type 2 direct cardiotoxicity. This is the prototype drug is trastuzumab, and it also it causes mostly reversible kind of cardiac dysfunction, but it also predisposes to further toxicities of other drugs, which, for example, aggravates anthracycline toxicity. But then we found out that the multitarget receptor tyrosine kinase inhibitors have another different set of, cardiotoxic effect. So finally, the summary is that the mechanism is not really known, and therefore the classifications kind of nonsense. So, for example, from Whitle's recent expert analysis, they said that this type 1 and type 2 cardiotoxicity is a complete nonsense because each drug has different mechanisms. So it's very important to see the mechanism of this toxicity. So we think that since there is no real classification, we can introduce this new term, hidden cardiotoxicity. So that means that the toxicity of a drug that manifests only in the diseased heart, for example, during ischemia reperfusion, or in the presence of its major comorbidities like diabetes, hyperlipidemia. But then we try to make a proof of concept study on it. So how can we prove that this theory is okay? So this, I will elaborate a little bit of this cardiotoxicity. So there's a drug-induced cardiotoxicity, which is a direct toxic signaling. You know, it's an easy to detect. Current safety testing does this. However, ischemic reperfusion injury might increase some deleterious cell signals, which may cause also myocardial cell death, and the same as risk factors like aging, diabetes, and so on. And these deleterious cell signaling can be, increased by the drug toxicity without having any direct effect, and that's what we call hidden cardiotoxicity. On the other hand, if the drug blocks the endogenous cardioprotective mechanisms, this might also have some unusual effect, which might manifest in the clinic. We don't know yet. So we planned a study accordingly, but first we were looking for a drug which has been withdrawn from the market. So we know already that it exerts hidden cardiotoxicity, since the cardiotoxic effect was revealed when the drug was on the clinical use. And for that, we used rofecoxib, which is a COX-2 inhibitor, and it has been removed from the market by the manufacturer because some trials showed that rofecoxib increased the occurrence of myocardial infarction. It also increased cardiovascular events, and it increased the risk of arrhythmias. So it means that rofecoxib had some ischemic cardiac events as an unexpected side effect. So we aim to set up a hidden cardiotoxicity testing platform where we examined if rofecoxib might interfere with ischemic reperfusion injury or cardioprotection by preconditioning, which is the most robust cardioprotective effect. So we designed a very, very simple study. So we fed the rats with for 20 days with vehicle, then rofecoxib in a clinically useful dose, and we applied to these rats myocardial coronary occlusion and reperfusion with or without ischemic preconditioning, and at the end we measured the myocardial infarct size, and of course we measured mortality of these rats. So what we have found is that rofecoxib increased mortality of due to ischemia reperfusion in these rats. However, ischemic preconditioning protected against this rofecoxib-induced increase in mortality. So we checked what is the reason of the increased mortality of rofecoxib, and we found that rofecoxib increases the incidence of irreversible ventricular fibrillation during ischemia reperfusion. So here you see the vehicle, and here you see the numbers of ischemia reperfusion when ventricular fibrillation occurred, usually at early times of ischemia. So this is the arrhythmia map with and without rofecoxib. You know, when there is black, that's already ventricular fibrillation. These are the number of animals in the certain groups. So what we saw is that rofecoxib basically killed these animals during ischemia when they were under ischemic condition. And how? So our collaborators in the pharmacology department in Szeged, they measured in isolated papillary muscle if there is any change in action potential duration. And yes, there was prolongation of action potential duration by increasing doses of rofecoxib. So we concluded that this pro-arrhythmic effect during ischemia reperfusion was due to prolongation, maybe due to prolongation of action potential duration. And next, these are the infarct size measurements, and this was very interesting. So while rofecoxib killed many of the rats due to ischemic arrhythmias, it decreased infarct size significantly, and also preconditioning decreased infarct size together with rofecoxib. So on the one hand, it had a pro-arrhythmic effect that killed the animals, and on the other hand, it seems that it's a cardioprotective. However, since we know that in the rofecoxib group there was much more mortality, we thought that only the survivors had less infarct size. So this result, this cardioprotection, would be a simple outcome of different mortalities. So then we went to isolated cardiac myocytes and subjected them to simulated ischemia and reperfusion and checked if rofecoxib itself has a cardiocytoprotective effect. And definitely it had a dose-dependent cardiocytoprotective effect. So here you see ischemia reperfusion induced change in the viability of these cells, and there was a dose-dependent improvement of and so decrease of cell death. So the conclusion so far is that hidden cardiotoxicity can be revealed by safety testing in the diseased heart. And in these very simple studies, what we did so far, that rofecoxib increases acute mortality due to irreversible ventricular fibrillation, which is probably due to the increased duration of action potentials in the heart. However, ischemic conditioning protects against rofecoxib-induced hidden cardiotoxicity because it blocked this action of rofecoxib. And this was the first demonstration what we just published in Cells, that the preclinical cardiac safety testing of drugs in this study, rofecoxib, in the presence of ischemia reperfusion may reveal hidden cardiotoxicity. So basically here we showed that rofecoxib, if it were tested in early preclinical phase in ischemia reperfusion injury, the developers might already knew that in ischemic patients it could be dangerous, as a meta-analysis showed that there was an increased arrhythmias, for example. So now we are developing a hidden tox testing platform to have a set of models where an early prediction of this unusual, unexpected mortality or unexpected side effect of drugs can be revealed. So what are the molecular mechanisms of hidden cardiotoxicity? These are still ongoing studies. What we do is, since we don't know any we don't have any clue, so we go for an unbiased microRNA, mRNA omics and multiomics analysis, and then we create a network, molecular network analysis. And I think also that this will be a future unbiased method to reveal new mechanisms. And these are ongoing studies. And I also want to advertise that the need for this network medicine is very important. We just published a paper. It's a worldwide alliance for network medicine, and we also developed some softwares to do the mRNA, microRNA interaction network. Another question is that this hidden cardiotoxicity can be revealed by adverse drug reaction reports, which is collected by the FDA and also the European Medicines Agency. So by this network approach, we also check this adverse event report. We developed the software to analyze it in a network analysis. And then we try to find out that this network analysis of these adverse event reports will give us a more prediction of true positive side effects of the drugs currently on the market. And here with this network approach, we could also show the very important connection with rofecoxib with acute myocardial infarction. So the 30 minutes is already done. So the final conclusion is that the importance of the cardiovascular comorbidities in preclinical drug development is evident, to test efficacy of cardioprotective drug candidates and also to reveal hidden cardiotoxicity of all drug candidates at an early preclinical phase. These are the group of who is involved in this hidden tox studies, and also collaborators in the hidden tox studies, and acknowledgement to these groups as mentioned. And as usually, the others also showed where they work. I have just some slides where we are. So our group is located in Budapest at the Semmelweis University on these floors. This is a basic medical science building. And our company is located in a high-tech science park, and also in Szeged in the historical center. Szeged is in southern Hungary, where vitamin C was discovered. And the next opportunity to come to Budapest is the FCVB meeting, the Frontiers in Cardiovascular Biology, which now, due to the COVID situation, moved to next year. And Budapest is really a beautiful touristic destination, and the venue will be at Semmelweis University. Our university is celebrating the 250th anniversary, and you know why even the recent Google picture, this guy is Ignaz Semmelweis, and unfortunately This guy was the one who found out that washing your hands inhibits spreading of diseases, and this became very actual now. So this will be in another basic science building of the university. There will be the venue of FCBB, and cardiovascular research will have a virtual session tomorrow where there will be an interview with me and the organizers why we moved it to next year. So thank you very much for your attention, and I'm happy to answer any questions, and especially in cardiotoxicity, I'm happy to learn from you. Thank you very much. I stop sharing now. Your microphone's on green.
Andre.
Sorry, I forgot to unmute. Thank you so much, Peter. That was very interesting. A lot of new data, new hypotheses, new concepts. Now it looks like cardioprotection gets more complex. We're getting, it gets to a higher level now. While people are, the attendees are busy writing some questions, maybe I'm going to ask with 1 or 2 questions. I was just thinking, the concept of hidden cardiotoxicity, when you have 2 cardioprotective drugs, or 2, for example, a cardioprotective drug that you add with conditioning, ischemic preconditioning, and when you lose the protection, do you think that one of the explanations could be that actually both of these drugs will stimulate the same signaling pathways and will overstimulate these protective signaling pathways that can actually eventually become toxic? Because we know that activation of free radicals, activation of AKT, activation of STAT3 will actually, overactivation may be deleterious. So is it something that could also explain this hidden cardiotoxicity when you add these 2 drugs?
Actually, to tell the truth, we don't have any idea of the mechanism of this hidden cardiotoxicity. Because if we had any ideas, we could just check that pathway if it's activated or not. So what we see now is that certain drugs might increase the the cardiocytotoxic effect of ischemia reperfusion injury, and some drugs might inhibit the effect of endogenous cardioprotection, like pre or postconditioning. So whether it is an overstimulation of the free radicals or what mechanisms, we really don't know. And looking at the literature, it's very surprising, but cardiotoxicity, the mechanisms of cardiotoxicity is kind of strange, that nobody knows what is exactly. There's no common mechanisms, it seems. So every drug has different mechanisms. We don't know, really, we don't know.
So my second question is, I was very intrigued about when you added rofecoxib with IPC, you reduce the infarct size, but then you mentioned that you actually increase the mortality. So now, did I understand correctly?
Yes, yes, it was very...
Your protection was based on the infarct size, but not on the mortality data. So I'm just a bit concerned there, because can we really talk about protection? And then it may raise the question, are we using the right endpoint by measuring infarct size with ischemic conditioning? Because
yeah. So these results were very surprising for us as well. So first of all, just again, you understood well. So rofecoxib killed more rats which were subjected to ischemia reperfusion than the vehicle, and this was due to ischemia reperfusion-induced arrhythmias. Okay. However, in the meantime, the surviving animals had less infarct size with rofecoxib than with the vehicle. So, and afterwards, since we put rofecoxib isolated cells subjected to ischemic reperfusion, and they protected ischemic cells. So we think that rofecoxib really have some cytoprotective effect, but in the meantime, they have a pro-arrhythmic effect, which manifests only during ischemia reperfusion. So if you check rofecoxib safety in a healthy tissue, you don't see anything. Even at high doses, it seems safe. But when you subject to ischemic reperfusion injury, it prolongs action potential and induces ventricular fibrillation and kills the animals. So if this had been done before, in the preclinical setting, the whole development would be more cautious of this COX-2 inhibitor. So,
but it was worse when rofecoxib was given with IPC compared to rofecoxib alone.
Yes, it means that IPC, so let's first discuss the arrhythmias. Preconditioning protected rofecoxib-induced pro-arrhythmic effect. So first of all, this was also surprising for us. So it seems that preconditioning can be cardioprotective against this pro-arrhythmic effect of rofecoxib. That was one thing. The other thing is the infarct size. Okay, so infarct size, rofecoxib by itself reduced the infarct size, and preconditioning together with rofecoxib further reduced the infarct size. So that means that rofecoxib did not inhibit preconditioning. On the other, moreover, it had an additive tissue protective effect. So,
but I guess, Peter, I mean, that kind of fits into the narrative of what you were saying, that you can't predict cardiotoxicity based on simply healthy animal data.
Yes.
That actually the mechanism for cardioprotection might be different from the mechanism for, let's say, induction of arrhythmias.
Yes, back to the question of, Sandrini, I didn't answer that. I think that for cardioprotection, we should measure infarct size, we should measure arrhythmias, and also post-MI myocardial function as well, because these three determines life on a long run. So, acute arrhythmias may kill the subject, and post-MI heart failure also has a lot of mortality. So, yeah.
Thank you, Peter. I think we're going to move with some questions from the attendees.
Yes. So how should we, you read the questions, yes, or I read the questions?
Can you see the question, Peter?
Yeah, okay, let's start with Vibarani, yes. So only the essence, natural products have gained significant attention in drug-induced cardiotoxicity. Have you tried combination therapy with natural products to combat cardiotoxicity induced by drugs? No, we haven't tried that. As I mentioned, we are really new in this field, so we are happy to receive any ideas and collaborators as well. So the first study that we done was with rofecoxib, and the current study that we are running is with sibutramine actually, so which is also a known hidden cardiotoxic compound. And because we want to further validate our systems, how to pick at the early preclinical stage these drugs. Of course, natural products are interesting. They can have very nice good and bad effects as well. So,
I mean, Peter, what is products? Sorry, do you mean like beetroot and stuff like that, or?
Yeah, that's what I think he or she asks. Yes, natural products. So I don't know. There is a lot of natural products. A lot of natural products are on the market as drugs. So
I would suggest the melatonin, of course.
Yes, yeah. So another question is that how the differentially expressed microRNAs identified in NGS. Could you please discuss it more? Yes, we are using for microRNAs, we are using PCR and DeepSeq analysis, and our own bioinformatic group identifies certain microRNAs in different species. And of course, we have to know the annotations for these microRNAs to define what is the expression level of them. And we also, it's very important to validate the differentially expressed microRNAs then with PCR, because the NGS is also not very reproducible, process. It depends on thresholds and so on. I'm not very much have everyday experience with an NGS. We do it with an NGS group because it's a very rapidly developing field. So it's better to go to specialized companies to do NGS, I think, or core facilities. So another Gabriella Stricciatella. The essence, given the positive effects of rofecoxib on ischemia, smaller infarct, have you tried to co-treat these animals with class 1 antiarrhythmic drugs like quinidine, disopyramide, procainamide, lidocaine, methylaxine, and so on to shorten the action potential and thereby overcome the pro-arrhythmic effect of rofecoxib? No, we haven't tried. It's a good idea, actually. We were, let's say, made some jokes in the lab that now we discovered the real cardioprotective drug, rofecoxib, which has been, removed from the market several years ago. Who knows? It definitely has an infarct size reducing effect, and it's also additive to preconditioning. So it's a good idea to try it with the combination with an antiarrhythmic drug. Yeah, we haven't done this so far. So, move to the next question. Michael Shetog. Given that rofecoxib prolongs action potential duration in aerobically perfused muscle, surely this is not a hidden cardiotoxicity. Surely it is overly dangerous. So, what we see again, that in an in vivo coronary occlusion reperfusion model, rofecoxib killed the animals, resulted in more mortality due to a higher incidence of irreversible ventricular fibrillation. Okay, so next we tried to follow up what would be the reason of this increased incidence of ventricular fibrillation. So it was obvious to see action potential duration because this is also done in normal safety testing of drugs. And what we have seen is that in the aerobically perfused muscle, in the aerobically perfused rat papillary muscles, we did not see any toxic effect, only when it was subjected to simulated ischemia reperfusion injury. So in the papillary muscle, we used this high lactate, low pH solution together with hypoxia, what we use usually in our cell culture models as well. So,
but Peter, did you see any prolongation of APD?
Only in the ischemic tissue,
not healthy hearts, nothing.
Healthy heart, nothing in the healthy heart, nothing. That was again a proof that this pro-arrhythmic effect is only seen when it's ischemic.
Because it seems to be the mic is suggesting that this drug actually does have a prolonging APD
yes, effect. Yes, it does have, but only
at least.
Yeah. Yes, but only when
just perfused muscle.
Yes, but not in the aerobically perfused muscle. So in the aerobically perfused muscle, there were no prolongation of action potential. Only with increasing dose of rofecoxib, it was a nice dose-dependent effect, actually. So the next question, David de Gonzalo Calvo. What about the potential cardioprotective effect of obstructive sleep apnea? That's a good question. To tell the truth, I haven't seen any clinical studies on it. In in general, if you make any organ, ischemic, that protects other ischemic organ. But of course, it's very important the duration of this ischemia fusion and maybe repetition. So in each conditioning model and the conditioning method should be set up precisely, because even some conditioning protocols, at least in preclinical trials, can be even dangerous. I think Catherine Prisklank's lab showed, for example, in diabetic mouse many years ago, that increased cycles of postconditioning was increased infarct size actually in this diabetic mouse, for example. So, but I don't know if the sleep apnea might be good. I don't know. Yes. Next question from Sina Hadipur. Has the data with rofecoxib been age stratified? For example, does rofecoxib have the same effect on young and old mice? We don't know. Here we used young and healthy rats subjected to ischemia reperfusion injury. So we don't know if this might be further affected by aging. But since aging is known to increase ischemic reperfusion injury and to inhibit, conditioning phenomenon, I might expect that in the old mice, rofecoxib could be even more toxic, but we haven't tried that yet.
On that note, Peter, you may also expect a difference between female and male.
Oh, yeah, yes.
All the experiments were done in male?
These studies were done in male rats only, yes. Yes, it would be also...
You may actually find something different in females.
Yes, of course, yes. This is another issue which is more and more promoted by, by our working group as well, that the sex difference should be taken into account. But we have been in a previous study that diabetes and the severity of diabetes is also different in different animal models as well. So it might, and the effect of diabetes on the heart in females and males.
Yeah, but I think the difference was mainly because there's already a difference in infarct size in male and female baseline. So whenever you have diabetes and conditioning, actually the female are protected already, so you can't protect them further, while the male can be protected because they were less protected baseline.
Yeah, it's also it's also difficult because in many of these diabetic models are different. So these are not the animal models. Unfortunately, are not very good for diabetes. So yeah. So the next question is Junhui San. The cardiotoxicity, such as increased nitration, could be prevented by co-treatment with the INOS inhibitor. Yes, of course, all kinds of, since we don't have any real idea what would be the mechanism of this cardiotoxicity of rofecoxib, so we don't go after a single hypothesis like INOS inhibition, because yes, definitely INOS inhibition could be okay. But since we don't have any proof that this would be a key factor, that's why we go for an unbiased exploration of the mechanism, and that's why we used the transcriptomics. So we measured all microRNA changes and all mRNA changes, and we built up a molecular network with network mathematics actually, and with that bioinformatic approach, I think that we can have a better chance to find something important mechanisms. Okay, so the next questions, only the essence is Caroline Archer. Given the difference in ion channel expression in rats, how translatable is this in human cells? Has this been tried in vitro? as this would be more amendable to early preclinical drug screening, bespoke investigation in disease-relevant human cells. You're absolutely right. You know, the rat is not a good arrhythmia model because the action potential duration is extremely short. So while in the humans, one action potential is a couple of hundred milliseconds, in the rats is only a couple of milliseconds. So we did not intend to find out any pro-arrhythmic effect. It was an absolutely unexpected finding of us. We thought that rofecoxib will interfere with pre-postconditioning or ischemia reperfusion injury. So, of course, the next would be a nice cell culture models in human cells and do ischemia reperfusion injury or engineered heart tissue, but this is not an easy task in humans. In fact, it's also necessary not only to check these drugs in ischemic reperfusion injury, but in multiple comorbidities like hyperlipidemia, diabetes, and ischemic reperfusion injury. But this needs a lot of further work. Actually, we just set up a new comorbidity model in vitro in myocardial cells with ischemic reperfusion injury and simulated hyperlipidemia. We just published it, I think, a couple of months ago.
Peter, I mean, this drug's been available for quite a while. Presumably, there must be data on this in patients, you know, that had MIs, etc.
Yeah.
You know, does that data reflect your findings?
Yeah, that's what. Yes, maybe in the beginning I told you that since we had this idea of hidden cardiotoxicity, we wanted to validate this idea. So we were looking for some drugs which is known to show this hidden cardiotoxicity. That is, it was revealed only in the clinical use, not before. And rofecoxib was a good choice because we knew that some meta-analysis showed more incidence of myocardial infarction, more incidence of ischemia-related arrhythmias, and more cardiovascular effects. The mechanisms, actually the company is still sued by a couple of trials still, although it has been withdrawn for many, many years. So I was also involved as an expert in one of these trials. So, okay. Next question from Michael Shattuck. When rofecoxib increased action potential duration in isolated muscle, what that in rat, and was it positively inotropic? Good question. We didn't check it. We did not. The isolated papillary muscle was done not in my group, but András Varró's group in Szeged, and they didn't measure inotropy, so contraction that with that model. So it was a very simple.
Mike corrects himself further down the questions. He says, apologies in terms of the protocol. So.
Oh, okay. So the next question is from Zoltán Hegyi. I'm just wondering if possible to identify hidden cardiotoxic effect on a drug in early phase. Will the researcher continue to develop a drug for clinical trial, or will stop testing a promising drug? That's yes, it's a good question, but any promising drug which shows, signs of safety problems, it should not further developed, or it should be developed in a way that you know what is the safety problem. So let's say if I was Merck at that time, and I knew our results before that it's pro-arrhythmic in ischemic patients, I would further maybe develop the drug, but I would put in the summary of product characteristics that in ischemic patients or in patients at risk of myocardial ischemia, it should not be used. It's contraindicated. So I think any knowledge on the preclinical development on the behavior of a compound in different comorbidities of the main indication is very important. So because you can better design clinical trials, because you know what will be the patient population you want to go for, and not all patients. You want to treat certain patients where you know that the drug is safe and effective. So, but this is the future. Now the clinical trials try to, you know, treat more the robust group of patients. Yutka Virag, are you also looking for known or even new biomarkers that would indicate severity and provide information not only about mechanism of action, but also structural property of these agents that may be driving the responses? I think that this hidden cardiotoxic idea, I think it will open up a new field of research, and when we will have more data on this comorbidity, the interference of comorbidity with cardiac safety and efficacy, of course, afterwards with bioinformatic methods, there could be some structural, you know, information drawn by using artificial intelligence, for example, what is necessary to use when you really dealing with very big data, like, for example, the drug adverse event reports. So yes, I think that more information will lead to this structural answers as well. Oh, yes. Then Rick Southworth. If I understand correctly, you are pushing the envelope to predispose your hearts to cardiac injury prior to testing cardiotoxicity, thereby making your screen more sensitive. Yes, absolutely. Presumably, the relevance of this approach depends on which pathways you are invoking with your prior injury, which may be more or less relevant to different comorbidities. Are you considering other sensitizing pathologies other than ischemia reperfusion to invoke other pathways? Yes, we do. We think that not only ischemia reperfusion injury, but simply, for example, hyperlipidemia on itself, diabetes on itself, and combined with ischemia reperfusion injury should be used. So these kind of testing platforms we are developing currently. What we call is now hidden tox platforms. Yes, I think it's very important in drug development.
But presumably then is for, you know, heart failure, for,
yes,
cardiorenal syndrome. I mean, you know, the sky's the limit here, really, isn't it?
Yeah, absolutely, yes. So Henrik Tuckmeyer, beta blockers raise myocardial glycogen levels. Is glycogen cardioprotective? I think it is. Oops, this is I should know, but I apologize. I don't know if glycogen is cardioprotective by itself.
I can't help
GSK. Yeah, beta inhibition was shown to disrupt cardioprotection. So, I don't know, but beta blockers raise myocardial glycogen levels. I don't know, but beta blockers are not cardioprotective in the clinical setting, so they do not reduce myocardial infarct size. So I don't know if any drug which might more effectively increase glycogen level might be cardioprotective. I don't know. I can't answer this question. For runners in ischemic exercise, it's definitely important to build up the glycogen level. So I like to run, so I know that. So Rio Juni. Next question. Do you know if there is any report on cardiotoxicity of SGLT2 inhibitors so far? I don't think so. SGLT2 inhibitors are, very promising drugs. They seem to be also cardiovascular protective effects. So far, I don't know anything about it, but it would be nice to try in this hidden tox models. So, of course, after we validated with some known drugs, we are going on with new drugs, which still thought to be very safe, actually, and try to find out if they're really safe in the different comorbidity models as well. But so far, clinical data doesn't show any cardiotoxicity of SGLT2 inhibitors. On the opposite, it now currently shows positive effect as a cardiovascular protective effect. So we are running out of time. I think we should go up to 7. This is, I think, yeah.
We had one more question.
One more question. Okay, to go. Okay.
I think it's coming. Very.
Okay. So, what all microRNAs you found in RGS, are they unique or mere 29? Yeah. Okay. So I intentionally did not show the actual microRNAs. Because it's we going to publish it, and maybe there will be some intellectual property protection as well. We have some microRNA patents for cardioprotection, so we might have some on cardiotoxicity as well. So I don't disclose currently on the certain microRNAs what we found. As I mentioned that we do full microRNA omics and build up molecular network analysis on that. Michael Shattuck again, and I think that's the last question. Yes, of course.
About the glycogen.
Yes, and ischemic preconditioning protocols lower glycogen levels prior to index ischemia and accelerates ATP depletion in index ischemia, but still cardioprotective. So Michael Shattuck answered the question, or the previous question of Henrik Takmeyer. So
there is one question in the chat from Mohammed Naveed. I don't know if you can
there is another chat. Oh, I see. Okay.
Yes.
Oops, I can't see it. Where?
Okay.
Can you read it? I can't see that question. Sorry.
I can read it for you. Mohammed asked if rofecoxib has been withdrawn. Why are we studying it now?
The reason, because we wanted to validate our idea that we can really predict with our preclinical model that this drug is cardiotoxic, hidden cardiotoxic, because the original problem was that rofecoxib was developed, put to the market, it was taken by thousands of patients, and finally meta-analysis and the clinical trials revealed that it is cardiotoxic. So now we checked if we could have shown this cardiotoxicity in the preclinical model, and yes, we just recently published it in Cells that yes, we can show that this hidden cardiotoxicity of certain drugs, and that was the first study of us. So that's why I told you that I want to learn together with you, because we are also beginner in this cardiotoxicity field. So we moved from cardioprotection to cardiotoxicity based on our results in comorbidities, actually, and co-medications. And then we realized that these comorbidities are very important, not only for cardioprotection testing, but also for cardiotoxicity testing as well. So that was the switch. So, and I'm very happy that to find out some new collaborators in the cardiotoxicity field. So, thank you very much.
Peter, thank you so much. That was really a fascinating talk, new ideas, and really thanks for taking the time to answer to all those questions and for giving this excellent talk.
Yeah, thank you very much.
Yeah.
Thank you both. Thank you, Peter. Really good stuff. I learned a lot, and Sandrine, thank you for sharing.
Thanks for
passion.
Thanks for ISHR again. Okay.
Yes.
Thank you very much. Great society. So thank you.
And thanks, Davo, again for taking the time to organize all these webinars.
It's a pleasure. It's a pleasure.
Thank
you.
you.
Have a nice day.
Thank you. Bye-bye.
Bye. Stay safe.
Thank you. Bye.